Morning, everybody. I'm Dave Kirn, co-founder and CEO of 4DMolecular Therapeutics. We're harnessing the power of directed evolution for targeted genetic medicines. And hopefully this will advance, but maybe not. This is not working. Is there another? There we go. Okay. So this is a summary slide of the company. We're boldly innovating to unlock the full potential of genetic medicines for countless patients. Our company is based in the San Francisco Bay Area. We're about 140 employees, and we have our own GMP manufacturing facilities and R&D facilities on site. Our platform is a Nobel Prize-winning technology called directed evolution. The starting point of directed evolution is to create massive genetic diversity of biologics that are not present in nature. So these are essentially synthetic. We start with on the order of 1 billion AAV synthetic capsids to start with. And then through directed evolution, we create targeted and evolved vectors for any specific disease or set of diseases we want to address. So one of the advantages of this in terms of our pipeline is it's very modular. Once we have a vector in hand, we can swap in and out different transgene payloads to treat a variety of diseases with the same vector. We have lead vectors in 3 therapeutic areas currently: ophthalmology, cardiology, and lung. And in clinical development, we currently have 5 clinical candidates currently in clinical trials. And our strategy is to become a fully integrated biopharmaceutical company. So this crowd needs no introduction to the difficulties with AAV. AAV obviously has great promise, but there have been a number of hurdles in the field, such as difficulty with delivery, transduction, and gene expression, as well as immunogenicity, pre-existing antibodies, and the like. So we felt that by using directed evolution, we had an opportunity to overcome these limitations in the field and move beyond specific rare diseases and move on to large market opportunities. So there are 3 steps in the directed evolution process we utilize. Number 1 is to create libraries of highly diverse capsids, again, that are synthetic and not seen in nature. So we have on the order of 1 billion of these. We start with a variety of different starting serotypes as well as vectors that we've discovered through this platform, and then induce diversity through a variety of molecular biology techniques. So now we have in the freezer at any given time a massively diverse set of capsids. Now what are the second and third steps? So step number 2 is we go to the whiteboard and we just ask a question for any disease we want to treat. What sort of vector do we want? What route of administration would we like to use? What dose range do we want to be in so we have a reasonable cost of goods and a good safety profile? Are we trying to target tissue broadly or specific cell types within a tissue? And then do we want to also build in relative resistance to antibodies in the population? So these are each part of what we call a target vector profile. Once we've defined that target vector profile, we then take the 3rd step, which is to do iterative competitive selection processes in non-human primates. Very important. With evolution, you get what you select for, and if you use a species that is not close to human, there's a high risk you're not going to get what you want. So in this case, we do everything in primates to make sure the immune response and the anatomy and the physiology is as close to humans as possible. So we do iterative rounds of this, and we funnel down from 1 billion vectors all the way down to a handful of highly effective vectors that match the target vector profile that we had. So as you can see in this diagram, we have 3 lead vectors, all of which are in lead products today. Okay, so back to the modularity concept. Once we have a vector in hand, we turn it over to our CMC group who can manufacture it, characterize it, and then we put it into human cell and disease models. These can be normal cell types that we're looking to target, or from patients, disease cell types where we can look at therapeutic effects in those. We also go back into primates for dose ranging and then file the IND and go into the clinic. And importantly, with this technology, we can deliver simple gene replacement, we can deliver and express approved biologics, and we can even do RNAi knockdown and gene editing. So it's a very robust and flexible platform. So this is our pipeline. These are all wholly owned assets across 3 therapeutic areas. Ophthalmology, our lead asset is 4D-150 for wet AMD and DME. That's currently in phase 1/2 trials. We We also have products for XLRP and choroideremia, rare diseases of the eye. 4310 is our IV low-dose Fabry disease product candidate. This is enriched for cardiac delivery as well as kidney delivery. We'll show you some data on that today. And then A101 is our aerosol lung vector for delivery for cystic fibrosis lung disease. And again, these are all currently in clinical trials. And all of these were internally invented with directed evolution at 4D. So we'll start with the ophthalmology franchise and 4D-150. But first, R100. So this is an intravitreal vector that we discovered and patented that allows us to use a simple intravitreal delivery method to target the entire breadth of the retina and all layers of the retina at doses which should give us a highly favorable safety profile. So to remind the audience, in the upper left, you can see the problem with wild-type conventional vectors. They didn't evolve to get through the inner limiting membrane and the other barriers to transfer from the vitreous into the retina, so they get bound up on that layer. And because of that, you can see in the upper right, many companies and academics have gone to the subretinal approach to try to transduce a small percentage of the retina. We took a different approach and evolved R100 for penetration through the retina. And in the lower right, you can see an in-life image from a primate looking at fluorescence from GFP from a delivered transgene. That oval area in the center, that's the fovea and macula right around that. So we get incredibly high expression there. But you can see we also see expression broadly out to the periphery as well. So 4D-150 for wet AMD and DME, you all know this is a large market with significant unmet need given the frequency of intravitreal injections required for most patients. There's a lack of adherence and often breakthrough symptoms. It's obviously a large and compelling market, and we chose to address this two ways. One is to express a aflibercept protein from the vector itself, and the second is to express an RNAi to target the VEGF-C. And there's excellent clinical data to say that that combination could be more effective than a aflibercept alone. So in a single gene medicine product, we're targeting 4 different molecular drivers of angiogenesis. This is an intravitreal product, as I said, and we're currently in phase 1/2 studies. This is the study design. It's a phase 1/2 dose exploration study. We start at what we believe will be a therapeutic dose level at 3E10 vector genomes per eye. So for those of you familiar with gene therapies of the eye, that's a very low starting dose, and we expect that we may be able to go down from there as well. So we built that into the study design that we can either go up or down in dose from the 3E10 based on the totality of the data. This is designed to enroll 12 to 15 patients during dose exploration, and then we roll immediately into a dose randomized phase 2 where we look at 2 different dose levels of 4D-150 in 20 patients each, and then a 10-patient aflibercept control arm. So exciting data coming down the pipeline from this study. So now on to cardiology. This utilizes our vector C102 for Fabry disease. So 4D-310 is a product for Fabry disease that has a very unique dual mechanism of action, very different from other standard protein approaches and gene therapy approaches. You can see the product design on the left. It utilizes C102, and then we express a codon-optimized GLA transgene, and we use a ubiquitous promoter because in Fabry disease we want expression throughout the body and all the key target organs. In terms of the vector itself, in the upper right, you can see the problem with conventional vectors, IV. If you want to try to deliver to the heart or skeletal muscle, you've got a lot of barriers in front of you, including the liver. So what we did is directed evolution to evolve a vector that could get past those barriers and give widespread transduction throughout the myocardium to directly transduce cardiomyocytes and express AGA in the heart cells themselves, which is the number one need in these patients. There's a high unmet medical need. ERT does not cross-correct the heart, and there's no evidence from animal models to date that you get effective cross-correction of cardiomyocytes, specifically from the blood. So very different approach. We also see clear transduction of podocytes within the glomeruli, which is, again, an important target in Fabry disease. So this is the study design for 4D-310. We use broad enrollment criteria, both classics and late onsets, males and females, and we also enroll whether or not patients have non-existing or low-level AGA antibody titers. Endpoints are safety, serum AGA activity, and then for cardiac, we look at both functional and imaging endpoints, including CPET, quality of life, MRI, and echocardiogram. And in the Asia-Pacific study, we'll also be doing cardiac biopsies. These are the first 3 patients that we've reported on in this study. These are all classics. 2 of them, patient 1 and 3, were both on ERT when they came into the study. 1 was off. And 2 of them had either low or mid-range AGA antibody titers. Patient number 2 had extremely high AGA antibody titers, and after this data, we've moved on and excluded these patients from further treatment. This is describing the cardiac imaging that we do on the study, both MRI and echo. In terms of the MRI, we can look at sphingolipid substrate accumulation in the heart using a native T1 imaging of the septum. Echocardiography with GLS is similar to a ejection fraction, so we can look at cardiac function by echo, and all of this is designed, monitored, and reviewed by the Duke Clinical Research Reading Center. So this is initial data on the left on cardiac MRI in the first patient that reached 12 months, and on the right is GLS. And you can see on the left upper panel, the MRI, we would have expected at 1 year for patients to get worse. That's that gray bar that you see below the line. That's historical data on ERT, and what you can see is a significant improvement in this patient's cardiac MRI, suggestive of clearance of substrate due to the direct transduction of cardiomyocytes and AGA expression in those cardiomyocytes. On the right upper panel, global longitudinal strain, we see again, we would have expected at 1 year with ERT for that to come down and worsen. Instead, what we saw is a significant improvement in the ejection fraction function. In both cases, these are well above the statistical noise of this assay, so they're felt to be statistically and clinically meaningful. So in summary from this program, 4D-310 is a unique dual mechanism of action relying on direct transduction and gene expression of AGA in cardiomyocytes and in podocytes within the kidney glomerulus. We've seen a nice manageable safety profile with no dose-limiting toxicities, and we have seen clinical activity both in terms of systemic AGA levels, manyfold above normal in the 2 patients with the lower levels of AGA antibody titers, and then promising early data on cardiac endpoints, which we'll update in the near future. And phase 1/2 enrollment is ongoing both in the US, Taiwan, and Australia. So to wrap up with our cystic fibrosis lung disease program, this utilizes our vector A101. You can see the design of the product on the left, utilizing a strong small promoter and expressing a truncated form of the CFTR, which lacks a regulatory region. This is a highly functional CFTR transgene product and has been validated in animals and in human cells. You can see in the upper right the problem with AAV delivery in lung of these patients is the vector seems to get bound up in the mucus barrier, and it also can be neutralized by antibodies that are present in that barrier. So we evolved A101 in primates to get through that mucus barrier and to be highly resistant to human IVIG pooled from many thousands of patients. This is data with 710 in primates looking at gene expression of the CFTR after an aerosol administration at a dose that's similar to our starting dose in humans, and you can see in brown staining in the lower row, widespread expression throughout the lung airway and the trachea, bronchi, and alveoli. This is the study design, again, dose exploration study starting at 1E15 VG's flat dose, followed by 2E15 in the second dose cohort, and then dose expansion. In this case, again, safety, plus we'll be looking at biopsies of the lung to confirm gene expression in the airway. And again, the target would be at least 15% is where we'd like to be to have clinical benefit. And we'll also be looking at percent predicted FEV1 over time. So with that, I'd like to thank you and just remind you that 4D is focused on directed evolution to unlock the full potential of genetic medicines. Thank you very much.